In Chapter 8, we're going to explore enzymes and look at the basic concepts and enzyme kinetics. So just to give an outline or overview of the chapter, we'll look at the enzyme overview, the definitions of enzymes, all in terms of that, and relate it back to some information from general chemistry. We'll look at thermodynamics. We'll talk about some relevant equations, review some of the variables that are involved. We'll look at enzyme reactions and discuss transition state theory. We'll talk about Michaelis-Menten kinetics and how that applies to enzymes, and then we'll discuss enzyme inhibition. So, about a quarter of the genes in the human genome will encode for enzymes, and nearly all known enzymes are proteins. Okay, so enzymes are proteins. And these proteins are effective because of their capacity to specifically bind with a wide range of molecules. And enzymes are going to serve as catalysts. And the way that they're going to catalyze reactions is by stabilizing the transition state. And if you remember back to general chemistry, we talked about transition state theory, or at least introduced it, and that transition state is the highest energy species in a reaction pathway. So it helps stabilize that and facilitates that reaction. In addition, we know that enzymes have specificity. That just means that they're able to bind or interact with very specific types of molecules. Now we'll see as we go through our study of biochemistry that many of the enzymes we encounter will require some level of cofactor, and there are several different types of substances that can serve as cofactors, which we'll explore in more detail in the future. So enzymes catalyze reactions. That means that they are going to accelerate reaction rates. And they can accelerate reactions by factors of as much as 1 million or more. So they really are going to have a significant impact on the rate at which the reaction takes place. So what we're looking at here, this table shows how long some common reactions would take if it weren't for the enzymes being present. And you can see that there's a significant change in the rate of the reaction once an enzyme is added. Many or most of the reactions present in biological systems would not take place at rates that would allow life to be sustained. So if we're looking here at some of these, we have the uncatalyzed reaction rate, so no enzyme, versus the catalyzed reaction rate. And you can see how slow these reactions occur in the uncatalyzed state versus the catalyzed state. So it's really telling us the activity of that particular substance or the function of that substance when the enzyme is present versus when it's not present. Now, enzymes are going to be highly specific in terms of reactants and substrates. Enzymes catalyze a single chemical reaction, but they can also catalyze a set of closely related reactions. So, for example, proteolytic enzymes will function to catalyze proteolysis. Now, proteolysis is hydrolysis of a peptide bond. So if we have a protein structure, we can break the peptide bonds that are present. Many of the proteolytic enzymes can also catalyze hydrolysis of an ester bond because these are similar types of reactions or hydrolysis reactions. So the proteolytic enzymes, they don't possess the high degree of specificity that many of the other enzymes do, but they're still going to function in a similar process. So we either have a very specific enzyme for substrate, or we have an enzyme that can work on a particular type of substrates. The specificity of an enzyme is due to the precise interaction of the substrate with the enzyme, so whatever is binding to it. And it is really the result of the three-dimensional structure of that enzyme protein. So as we've mentioned, many enzymes are going to require cofactors in order to function. And the cofactors are going to be small molecules or metal ions that will allow the reaction to take place. Now, those that actually require these cofactors, when we don't have the cofactor present, we call this the apoenzyme. Okay, so the apoenzyme is going to be the enzyme without cofactor. In the presence of the cofactor, when it is there, we're going to call that the holoenzyme. So this is when the enzyme, you have enzyme plus cofactor. The whole thing is present. So the apoenzyme plus the cofactor gives us the holoenzyme. Now the holoenzyme is a complete catalytically active form of the enzyme. So this is going to be catalytically active. And so what we're looking at in this table is just a variety of different substances that can serve as cofactors. And cofactors will come in one of two forms. So we see here that we have coenzymes and we have metal ions. Okay, coenzymes and metal ions. And the coenzymes are small organic molecules. Coenzymes are often vitamin derivatives, and they can be tightly or loosely bound to the enzyme. If they're tightly bound portions, we refer to them as prosthetic groups. So you'll hear that term throughout our study. The weaker bound cofactors are considered co-substrates. So strongly bound are prosthetic groups, weaker associated are co-substrates. These cofactors will differ from substrates in that the same coenzyme can be used by a variety of different types of enzymes. So one substance doesn't only work as a coenzyme for one particular reaction, it can serve as a coenzyme for, or a cofactor for, many different reactions. Enzymes are going to play a vital role in energy transformation. We'll see this play out as we explore the types of biochemical processes throughout the semester. But just know that oftentimes when we encounter enzymes, we're going to see some molecule associated with them, which may be these coenzymes, or we'll see metal ions that need to be present in order for them to function. So this is just a list of some of these coenzymes and metals, and then some of the enzymes that they're going to interact with. So if we explore thermodynamics, there are a couple of things that we know to be true from our study and other courses. First, we know that a reaction can take place spontaneously only if the free energy is negative, or if it's an exergonic reaction. So the spontaneous reaction has a negative delta G. If a system is at equilibrium, the free energy is 0 and there's no change that's observed. Kind of implied in this first statement here is that if we have a positive free energy, then that reaction can't take place spontaneously. It has to have energy input. The free energy change will depend on the free energy of the products minus the free energy of the reactants. And you've done this calculation many, many times in your general chemistry courses. The free energy does not indicate the rate at which the reaction occurs, and that's important. Free energy just tells us whether or not it's going to occur spontaneously. It doesn't tell us how much time it's going to take in order for the reaction to occur. We can determine the rate free energy of any reaction given some of the information that's shown here. So if we just have this overall generic reaction, A plus B produces C plus D, the free energy change for this particular reaction is equal to the free energy of the standard state. Now this value here we would actually obtain by using this type of relationship, the products minus the reactants, plus R, which is that constant, the 8.314 joules per mole Kelvin. T is your temperature, and that temperature is going to be in Kelvin. And then the ln of this ratio of concentrations of products and reactants, or Q. And if we're at equilibrium, this represents K. So we've seen this as Q or K in our study of thermodynamics in the past. And that is just either the reaction quotient or the equilibrium constant. So we can look at this in a little bit more detail, simplify the equation, allows us to look at equilibrium conditions. We can explore the various terms that are here. Now we would need to calculate free energy in the standard state before calculating the overall free energy of a substance. So remember that the equilibrium constant, K, is that ratio of products over reactants in terms of their concentration. You can do some rearranging or substituting, and we see that the delta G is equal to negative RT ln of K. Now, depending on what information you're given in the problem, you can rearrange this expression. How do you get rid of an ln? Well, you take e to that value. So you can do some rearrangement here, and that may allow you to calculate for K equilibrium. Now, you may have K and be looking for some of the other variables. Now let's look at a calculation using this information. So let's say we want to look at the reaction where we take DHAP, which is dihydroxyacetone phosphate, and we convert that to GAP, which is glyceraldehyde 3-phosphate. And we will see this reaction throughout our study this semester. Now, let's say that the ratio of the product over reactant, the GAP to DHAP, is going to be 0.0475. And this is at 298 Kelvin, and also pH of 7 because we want it under physiological conditions. Now this piece represents K. That's my K value. So we can calculate the delta G under the standard conditions for this by looking at that equation, negative RT ln of K. So we're plugging the information that we have into this equation. So we have negative 8.314, and this is joules per mole Kelvin, times the temperature, which is 298 Kelvin, times the ln of 0.0475. And this gives us a value of 7,549.2 joules per mole. Now, just as an aside, delta G is typically reported in kilojoules, so you would want to convert this to kilojoules, and that would give you 7.55 kilojoules per mole. So that's just showing how we can actually calculate information here. And we can apply this idea to any system. That will allow us to see whether or not it's a spontaneous reaction. Whether that free energy of the system is large or small, the standard free energy, that delta G with the little degree sign, that is going to be dependent upon the equilibrium constant, or the concentration of reactants and products. And this table is showing us the relationship between that equilibrium constant and that delta G. So we see that as K increases, the delta G becomes negative, or the reaction is going to become more spontaneous or more likely to occur. Remember that negative delta G will indicate spontaneous reactions. So as our K is getting bigger, we see more negative delta G's here, which means more spontaneous reactions. So we've been talking about thermodynamics, but it's important to remember that enzymes are not going to alter the thermodynamics of a reaction. And they're also not going to shift the equilibrium state. Remember that delta G just tells us, is it going to happen, not how quickly it's going to happen. So enzymes are not going to alter the amount of products formed in a reaction. And they don't change the amount that you get, they just change how quickly you get to that. So the enzyme, it's going to produce the amount of product at a much quicker pace. So here's a graph, and what we're seeing in this graph, we have time plotted against the amount of product, so how long it takes and the amount of product there. And there's a maximum amount of product that the system approaches, and as the system reaches equilibrium, we kind of level off here, our curve kind of levels off. Okay? We're not going to make more product. So this graph shows that the process occurs on a second's time scale in the presence of an enzyme, but on an hour time scale in the absence of an enzyme. So we can see that here this reaction is occurring. We have no enzyme present, It's really, really slow and takes a really long time to reach or start to approach the total amount of product. Whereas when we do have the enzyme present, it pretty quickly within seconds, starts to approach that maximum amount. So the take home is that the enzymes accelerate the attainment of equilibrium, but they don't shift the equilibrium position. So we've mentioned that enzymes are going to function by stabilizing the transition state. So what does that mean? Well, when products are formed from reactants, they go through what's referred to as a transition state. And this is a state that has higher free energy than either the reactant or products. Reactants are referred to as substrates in the enzyme process. So we have our substrate or our reactant and our product here, and the transition state is that higher energy state. So we're going through this process where we have the substrate, and that's converted to our transition state, and then it's converted into product. So the transition state is a substance that is no longer reactant, but not quite yet product. And the energy difference between the substrate and the transition state is referred to as the activation energy. Okay, so this is the activation energy that we have, that distance between it. What enzymes do is they function to lower that activation energy. Okay, they're lowering the activation energy. Another way to state it is that the enzymes are going to facilitate the formation of the transition state. So they accelerate reactions by decreasing that activation energy. So uncatalyzed, there's my activation energy, this larger hump that I have to get over. Whereas when it's catalyzed, it's a smaller hump, a lower delta G. So key to the idea of enzymes and catalysis is this formation of the transition state, and how much energy does it take to form that transition state. Now, talking about catalyzed reactions, you have your substrate and you have your enzyme present. In order for this process to occur, you have to have the formation of an enzyme substrate complex in order for the enzyme to function. So that enzyme has to interact with the substrate in some way. We have been able to study the formation of enzyme substrate complexes, and there is some evidence for their existence. First, the reaction rate increases with increasing substrate concentration until there's some maximum that's reached. So you can continue, some experiments were done where they were looking at reaction rate and when we're putting more substrate in there, and there is going to be a maximum that's going to be reached. And the presence of a maximum would indicate that something is going to dictate or limit the overall rate of the reaction. There has been spectroscopic characterization that's been performed. So you can actually spectroscopically study the substrate in the presence and absence of the enzyme, and vice versa. And you can monitor how the changes occur within that substance. So you can see the structure of the substrate when it's bound with enzyme versus unbound with enzyme. And there's some X-ray crystallography data that's been used to look at the structural differences in that enzyme and substrate upon binding. So here we're just kind of highlighting that. There's some constant concentration of enzymes. If you have a constant concentration, the reaction rate increases. Okay, there's increasing with increasing substrate concentration until you reach some sort of maximum. And again, spectroscopic characteristics can monitor these changes, and X-ray crystallography can look at the actual structures that are present there. So we often refer to where that enzyme and substrate, that binding portion, as that active site, okay, where that enzyme is going to bind the substrate. And there are some common features that exist for active sites. First, the active site, it's actually a three-dimensional cleft or crevice. And that cleft is the result of different parts of the amino acid sequence interacting and coming together. The active site takes up a small part of the volume of the enzyme. So in terms of total volume, only a small portion of the amino acid residues of the enzyme will actually interact with the active site. It should be noted that without the overall enzyme, it wouldn't function properly, but it's only a small number of residues that actually take place. There's about a minimum of 100 residues needed to display enzymatic activity. Active sites are unique microenvironments. So in terms of the microenvironment, the active sites, they'll tend to be nonpolar in nature and exclude water. That will allow for specific types of interactions. The cleft itself may also have some residues in there which will aid or help in binding of substrate. Substrates are bound to enzymes by multiple weak attractions. Okay, so they're interacting with one another through weak intermolecular forces to form that enzyme substrate complex. And it's important that we have these weak attractions here, because remember that we don't want that enzyme to be consumed by the reaction. It needs to be regenerated. So we don't want any strong binding that would alter that enzyme. We want it to be able to release. And one statement that was cut off from the slide is that the specificity of binding will depend on the precisely defined arrangement of atoms within the active site. Enzymes are going to tend to bind according to one of two models. And you've heard of these models, the induced fit model or the lock and key model. In either case, there are specific features that the substrate must have in order to bind with that specific enzyme. So here in this figure, we're just showing the small portion of the overall enzyme, which is actually involved in forming the active site to bind the enzyme. So the highlighted residues are those that are going to be involved in that active site and binding. The remaining portions of the protein structure really are not directly associated with the substrate. They're important for the enzyme to function, but they don't bind that substrate. Here we're seeing an example of some of these intermolecular forces that are present within the active site. We'll explore more about the interactions that occur in active sites in the future, but this is just showing us some of them. And more specifically, in this case, hydrogen bonds are occurring between these. It's not the only bond that forms, but it is an intermolecular interaction that can occur between substrate and enzyme. So again, as mentioned, binding models, there are two, the lock and key and the induced fit, and we can go through and explore these. Just reviewing these definitions that you've probably heard in some of your biology courses. In the lock and key model, the enzyme and substrate molecules have very specific and complementary shapes. The shape of the substrate must match exactly to the shape of the active site, and we see that here. This shape has to match exactly to the substrate shape in order for them to come together and form the enzyme substrate complex. Whereas in the induced fit model, once the substrate interacts with the enzyme, then that active site is going to adjust or fit to that substrate. So they don't have to match exactly. They have to fit in one another, but they don't have to match exactly here. So you see that this pocket of this particular enzyme is not necessarily complementary or doesn't really match the shape of the substrate. But when the substrate comes into this pocket, you see that the active site kind of changes and fits to that substrate, producing that enzyme substrate complex. So we've defined enzymes, we've looked at what they mean, what the active site is, importance of transition state, formation of that enzyme substrate complex. But now we want to get into studying the kinetics of the enzymes. And one way that we study enzyme kinetics is through the use of the Michaelis-Menten model. Okay? So we're going to look at what this model is and what it tells us. This model allows us to examine rate versus substrate concentration. And this model provides us with an equation that we can use to study how enzymes are going to behave overall. The equation was derived based on experimental data. Now, many of the reactions that we're going to study are going to be referred to as bimolecular reactions. That just means that they have two reactants that are present. When that happens, their rates can be expressed, V is their rate, K, or the rate constant, times the concentration of the first substrate, or first substance, times the concentration of the second. This is a second order reaction. So remember, in general chemistry, we focus on zero, first, and second order reactions. So for a second order reaction, the units of that K, or for that rate constant, are going to be inverse molar, inverse seconds. Now, in order to go through and derive the Michaelis-Menten equation and figure out how kinetics is governed, there were some experiments that were done. So one of the ways we can study rates of reaction is by constructing a plot of velocity or rate versus substrate concentration. And we can do this by placing an enzyme in a beaker with an initial concentration of substrate equal to 0. So your initial velocity is 0. There's no substrate, it's not going to function. We can add an amount of substrate and measure the corresponding velocity. And we continue to incrementally increase the amount of substrate and note how the velocity changes. And we do this until we start to observe a curve. Now the curve will initially start to resemble a straight line, and then when we add substrate, and then the rate becomes directly proportional to the substrate concentration. So eventually you're going to end up with the slope changing and the curve leveling off as we reach some maximal velocity. So some maximum rate of enzyme activity. So this is kind of showing that you have your initial, this is one particular substrate concentration in there, and we see what's the maximum point that it can reach in terms of product formation. And then we put a little bit more in, we measure that concentration, and we measure that product formation. And so you start to see over time, these straight lines are starting to form little curves, and eventually you're going to reach some maximum where that curve is going to level off. So eventually you're going to get that leveling. That's the maximum rate of enzyme activity. Now we actually will never reach the Vmax, or the maximum rate, but we come close to it, or we approach it. So this is just kind of showing that idea there, where this is just plotting the same information in a couple of different ways. So if we look at this Michaelis-Menten kinetics, let's use this equation here in order to describe what's going on in this. So you have your enzyme plus substrate, and it forms some enzyme-substrate complex. But then remember, ultimately you want to form product and regenerate your enzyme. Okay? So enzyme plus substrate form the enzyme-substrate complex. So now I've drawn the arrows here just as arrows over, but remember that these are equilibrium processes that occur. So at each step, you can go in either direction. And that just means that as you have these coming together to form enzyme substrate, this enzyme substrate can also go back to forming enzyme plus substrate, but it can also go on to form enzyme plus product. So if we think about the kinetics that's happening and these equilibrium lines that are present, and you can go through the derivation that's outlined in the textbook, but overall you're going to come up with the Michaelis-Menten equation. And the Michaelis-Menten equation is this V naught is equal to Vmax times substrate concentration over substrate concentration plus Km. So here V naught is initial velocity. Of course, the Vmax is the maximum velocity. Km is the Michaelis constant. And that Michaelis-Menten constant, that Km, is equal to the substrate concentration at which the reaction rate is half the maximum value. Okay, and we're seeing that here. So this is half the maximum value. What is that substrate concentration? Okay. So in other words, if half the enzymes are occupied by substrate, they'll be functioning at half velocity. And then, of course, your S is your substrate concentration. So this is just taking that data that we previously obtained by plotting that velocity and substrate concentration and defining these terms. So you have some maximum that you never really reach, but you approach as your curve is leveling off there. That's your Vmax here, your Vmax over 2, or the velocity at half that substrate, gives rise to that Michaelis-Menten constant. So now how do you determine Km and Vmax values? Well, we can determine these values for any system by constructing what's called a Lineweaver-Burk plot. Okay, this is a double reciprocal plot. The method is necessary, especially for finding the Vmax, because using the Michaelis-Menten equation and plotting it, we approach Vmax, but we never reach it, so we can never obtain that. So we create this double reciprocal plot, and it's derived from taking the reciprocal of both sides of the Michaelis-Menten equation, so 1 over each side. So in this plot, the y-intercept is going to give us the value of 1 over Vmax. Okay, so 1, there's our intercept, 1 over Vmax. So if we know this value, we can solve for that Vmax number. The slope here is going to be your Km, your Michaelis constant, divided by your Vmax. And then your x-intercept is going to be 1 over, or rather negative 1 over the Michaelis constant. So if you know these values, the x-intercept, the y-intercept, and the slope, then you can find this other information. And actually, these plots, we'll see they're going to be important, these double reciprocal plots, when we're talking about enzyme inhibition here shortly. But the variations in the Michaelis constant can have physiological consequences, so it's important to be able to study or know that value. So we have seen variety in terms of the ability of enzymes to increase reaction rates. So it's not like all enzymes increase reaction rates by this amount. There is going to be some variety in terms of how much they increase the reaction rates. The rates of enzyme catalyzed reactions will vary for varying systems, and as those are varying, so will the Km values, those Michaelis constant values. So that Km is going to depend on the substrate and the environmental conditions. So things like pH, temperature, ionic strength, those will all impact what that Km value is going to be. The Km provides us with information about the substrate concentration that's required in order for significant catalysis to take place. So how much substrate do we need in order to get an observable change in terms of the rate? So this table is just illustrating a variety of enzymes. It's indicating the substrates that are used for those enzymes and showing how those K values are different for these. So do note, though, that these values will vary, and how they vary depends on the enzyme and substrate that's there. When we're thinking about the effectiveness of enzymes, we can use something called the turnover number. So this table is representing the turnover number. This is going to be the number of substrate molecules converted into product by an enzyme molecule in a unit of time, and that occurs when the enzyme is fully saturated with substrate. So again, it's turnover number, it's the number of substrate molecules that are converted to product by a particular enzyme per unit of time, and that condition is that the enzyme has to be fully saturated with the substrate. Now we often use a value called kcat in order to describe this, to represent this value, and that is equal to Vmax over enzyme concentration associated with some time. So here we're looking at the enzyme and just the turnover, the typical turnover that is expressed there. So this is going to allow us to determine the overall efficiency of the enzyme, because the overall efficiency can be calculated by looking at this kcat value divided by that Michaelis constant value. So looking at the numbers that are here, the higher the numbers, the more efficient the enzyme is. So something like carbonic anhydrase 600,000 is the turnover number per second. So 600,000 substrate molecules converted to product per second when we have that fully substrated. That's a lot of product. So we have this model that allows us to study enzymes and enzyme kinetics, but many of the enzyme reactions that we're looking at actually involve more than one overall step. And the multi-step reactions can be expressed through one of two different models. So we can look at different models to describe behavior, which are kind of a little bit more complicated than just simply saying enzyme plus substrate produces enzyme substrate complex produces enzyme plus product. So the two models that we look at, we have the sequential model, or sequential reactions, and then we have the double displacement, which is also called the ping pong model. So if we look at the sequential first, in the sequential reactions, the defining property here is that all of the substrate must bind to the active site before the enzyme can convert them into products. So everything comes in and binds, and then the enzyme is going to convert it to products. Now this particular model can be broken down into two subcategories. We can further classify it. It can be ordered, or it can be random. The ordered sequential model involves the substrate molecules binding to the active site in a specific order, and then the products would be released in a specific order. So in other words, if we have our little active site here and we have our substrates, let's say A and B, in the order, A would come in and bind, then B would come in and bind. And if A and B are converted to C and D, then coming out of the site, so when we're binding, A comes in first, then B comes in, then let's say that C would have to be released, and then D would have to be released. So there's a specific order that things come in and bind, and that things are released. Whereas in this random, if we're looking at the random model, the order, it's not going to matter, and the order of release of product doesn't matter. It is completely random. So doesn't matter what the order is. A can come in first, or B can come in first. C can be released first, or D can be released first. Makes no difference. Now in this double displacement reaction, the ping pong model, the first substrate binds and changes the enzyme to product. An enzyme substrate intermediate complex is formed. So looking at the same reaction, what does this mean? Well, if here is my active site, okay, let's say A comes in, and once inside, if A is our substrate, I'm going to form, this is going to give me an enzyme substrate complex, and that enzyme substrate complex, let's say EA, because it's substrate A that's binding to it. Then within there, this is going to be converted to enzyme plus product. So let's say product C is given off. And then C would be released. After that takes place, okay, once you have that first substrate being converted to product, then your second substrate, your B, would be able to come in. It would form another enzyme substrate complex and form the other product and then release that product. So two different ways to deal with multi-step reactions, sequential and double displacement. Now it's worth noting that allosteric enzymes, these are enzymes that contain multiple subunits and multiple active sites. They do not obey Michaelis-Menten kinetics. These enzymes will exhibit a sigmoidal curve, and we see that here with respect to plotting the velocity and substrate concentration. The shape is due to something called cooperative binding that takes place within these, and we will explore this in more detail in the future while looking at specific enzyme or protein systems. So now let's get into inhibition of enzymes. So enzymes can be inhibited, and this comes in a couple of different forms. We can have reversible inhibition or irreversible. Reversible inhibitors can be further categorized, and those can be competitive, uncompetitive, and non-competitive. Okay, so the reversible, competitive, uncompetitive, and non-competitive. Irreversible inhibitors dissociate very slowly from target enzymes. Some are bound very tightly, and so those are kind of more damaging. An example of an irreversible inhibitor is something like penicillin. So here what we're looking at are the reversible inhibitors. We have the competitive inhibitor, the uncompetitive inhibitor, and the non-competitive. So in the competitive inhibitor, they bind the same site as the same active site as the substrate. So if this is the substrate that comes in and binds that pocket, the inhibitor will come in and bind that same pocket and will prevent the substrate from being able to go down in there. With the uncompetitive, they bind a site that's only created when the substrate binds, and that will decrease the total number of enzymes available to bind substrate, because what it does is it prevents the release of what's inside that pocket. Okay, so it increases the affinity of the enzyme for the substrate. So in these uncompetitive, your substrate comes in and then the inhibitor binds, and that inhibitor binding is going to make this enzyme hold on to that substrate more tightly, and it won't release the product. Now in non-competitive, this one over here, this is going to bind whether or not the substrate is already bound. It can bind either the enzyme itself or the enzyme substrate complex, but it binds somewhere other than that active site. And we can explore each type of inhibition, competitive, uncompetitive, and non-competitive, in terms of what it does to the overall kinetics of our system, because it is going to change those properties that were observed through Michaelis-Menten. And so this is just we'll look at a plot of each of these, the rate versus the substrate concentration. When there's no inhibitor and our enzyme is functioning normally, is this line here, which I'll make red instead of green so we can differentiate. And so we can see when we're inhibiting it, how it alters that overall curve. Same is true here. Again, relative rate, substrate concentration. This is our normal enzyme, no inhibitor, and how is that altered in the uncompetitive. And then non-competitive, there's our normal uninhibited, and then what's going on down here. So you can study these different models and think about how the substrate concentration is going to be impacted. But what's going to allow us to really differentiate between the inhibitors will be the double reciprocal plots we talked about. So we can differentiate between inhibitors using double reciprocal plots, and it tells us about the information that we really observed in those previous graphs. So if you create these double reciprocal plots, you plot your original uninhibited enzyme, and then you plot the inhibited enzyme. When we have a competitive, when you have your competitive inhibitor present, the slope is going to be greater. So the slope of this line is greater. The y-intercept, you'll see, stays the same. There's no change in that y-intercept. But your x-intercept is going to change, and it becomes less negative through here. The competitive inhibitor, remember, it binds in the same site, same active site as that substrate. And because it binds reversibly, we can actually increase the concentration of substrate to bind and kick out that inhibitor. Because we can do that, the Vmax isn't going to change. Okay, so we're not going to see a change in the Vmax. Now the Km value is changed, and that's because the affinity of the substrate for the active site decreases in the presence of the inhibitor. So because the Km changes, the x-intercept changes. Because the Km changes, that slope is going to change. But because the Vmax doesn't change, that's why this intercept does not change. When we have this case of the uncompetitive inhibitor, basically what we're doing is we're just shifting our overall line, okay? No inhibitor present. It's the black versus inhibitor. So we're just shifting this up. So the slope itself doesn't change, but the x and y intercepts will change. So we see a change in the y-intercept and the x-intercept. Uncompetitive inhibitors bind a site that's only created when the substrate binds. So it decreases the total number of enzymes available to bind substrate because it prevents the release. Okay. So this is going to change the Vmax. The Km will decrease when that substrate binds. So if Km changes, you're going to see a change in the intercept that's present there. Our Vmax changes, so you see that change in the intercept. But the slope remains the same because those changes in Km and Vmax, they're kind of correlated to one another. So it's the same amount that they're going to be altered. Now with the non-competitive that we see over here, again, here's no inhibitor and inhibitor present. In the presence of the inhibitor, your slope's going to change. Okay, the y-intercept changes. And then you're going to see that your x-intercept is going to stay the same. So our y-intercept changes, x-intercept is the same, but our slope changes. Okay. Non-competitive inhibitors will bind whether or not the substrate is already bound. They can bind enzyme itself or enzyme substrates. We are going to see a change in Vmax. Vmax will be smaller. That's why our y-intercept is going to change. Because our y-intercept is changing, our slope is going to change. There are lots of different things that can serve as inhibitors. Compounds that resemble the transition state will be effective inhibitors because they're able to interact in much of the same way that your actual substrate would interact. So we did a lot of things, a lot of information was thrown at you in this video. So you do want to go back, think about those definitions of enzymes, think about the different models that exist, what's Michaelis-Menten model, how are enzymes and substrates bound together, what kind of inhibitors do we observe.